Good morning, everyone. Thank you so much for joining. We're going to wait another minute or two to allow others to join into the session before starting, but thanks for being here. All right, good morning, everyone. My name is Allison Garcia. I am a contractor from Publicis Sapient, and our team supports the Biomedical Research Informatics Computing System, or as we refer to as BRICS. And we are very excited to have everyone join us this morning for this working group seminar to learn about all of the capabilities that BRICS has, to also hear from you all in the community, to hear what is of interest of you in terms of the capabilities that the BRICS platform has and how we can continue to evolve for future needs of the community at large. So with that, I just want to kick off today's meeting with a few logistics. Next slide, please. Okay, perfect. So please keep your microphones muted. You can see many of the presenters who are part of today's seminar currently have their video on, but feel free to turn your video on during question time. But please, for the proceeding, please keep your video off and keep your microphones muted. This seminar is being recorded and it will be posted at a future date to the BRICS public website at the URL that's listed on the slide. There will be one break today, just one 15-minute break, basically scheduled halfway through today's session. Today's session is from 9 till noon, and we're anticipating that break to be around 10:20 this morning. We definitely want this to be interactive. We encourage you all to be active participants today. There is the chat, so the chat will be monitored throughout today's session. So please feel free to add questions or comments to the chat throughout the session. And then there will also be opportunities after each of the speakers present today for a short period of time for Q&A. And then there will also be time reserved at the end of today's seminar also for you to ask any questions you may have. Next slide, please. We are very excited to present the agenda for today's session. I am kicking it off. I will then introduce Dr. Matt McAuliffe, who is really leading this initiative here at CIT. But we have a series of speakers who are coming today to speak about various topics related to how the BRICS platform is able to support your needs in clinical research, as well as some of the specific capabilities of the platform, use cases for how the platform is currently being used. And also, we would love to get your feedback and have an opportunity to speak with all of you throughout the day at the end of each speaker and also at closing. Next slide, please. So I have the honor of introducing Dr. Matt McAuliffe. Dr. McAuliffe has been at NIH since 1998 and currently is serving as the chief of the Scientific Applications Services section in the Office of Scientific Computing Services here at the Center for Information Technology. Matt has his PhD in biomedical engineering, and his passion is to advance and empower scientific imaging research, as well as the ability to share biomedical research data in support of NIH intramural and extramural research initiatives. Matt is the creator of the product that is called the Medical Image Processing Analysis and Visualization Application, or MIPAV. And you can find some more information about MIPAV by clicking the link on the slide. But really, Matt's passion over the last decade that I've had the privilege of working with Matt is really to support FAIR, making data findable, accessible, interoperable, and reusable, and has developed the BRICS platform to really be an opportunity to serve the community to enable fairness in your research projects and communities. So with that, I will turn it over to Dr. McAuliffe to kick off today's seminar. Thank you.
Great, Allison, thank you very much. It's a pleasure to have everyone join today, and I'm going to kick it off by just giving a quick brief overview of the BRICS system, and I'll be delving into it a little bit more deeply in my talk, but I just wanted to lay some of the groundwork, and then we're going to, right after me, we're going to follow with Dr. Dominic Nathan, who's going to go and talk a lot about his work with the platform. Okay. There we go. So you can find out a lot about BRICS by just going to the website, which is just brics.cit.nih.gov. We have a lot of important information there at the site. We keep the site up to date. And for example, we have release notes as well as some of our collaborators that we work with and links to their particular instances of the BRICS platform. So just to give you a little historical information, BRICS was kind of an outgrowth of NDAR, which is the National Database for Autism Research. When we did the one-off system for NDAR, we handed that off to NIMH, and the DoD and NINDS had learned about the BRICS or about an NDAR, and they wanted a system similar to that. So we discussed a design for the BRICS platform, which is a generic platform that can be deployed, easily deployed in a number of different ways. And so I take input. I'm members of a number of working group committees, the FAIR working groups, technical integration working groups, all these different kind of working groups here at NIH. BMIC is another one. And I try to extract important kind of use cases from those working groups, as well as the stakeholders within our repositories, to enhance the system to meet everyone's needs. So we've really done a lot of work on getting data into the system and being able to quickly deploy new versions of the application. BRICS is an open source system. We have access to all the source code. We don't have to worry about trying to get transfer agreements or anything like that. We can add the functionality immediately into the system. And on the right-hand side is a list of some of the applications that are part of our instances of BRICS. Okay, so we have over 300 research studies within the instances of BRICS, over 300,000 research participants, as well as 7 million plus records, and that's growing every year substantially. I did want to talk a little bit about fabrics, meshes, and data lakes and how that relates to BRICS. And it seems to be a rather hot topic now. And at least, I mean, I don't know that there's consensus on exactly what these definitions actually mean, but this is at least what it means to me, is a data fabric is more of a single instance where you have a complete system that addresses all the needs of that particular stakeholder community. And so you could actually deploy a single instance of BRICS because it already has so much functionality, privacy preserving record linkage, the GUID system. We have data fabric. We have the repository, as well as common data elements. We support different common data models within that common data element effort. But we can also, and now we're working on trying to fit into an ecosystem or a data mesh, where you have multiple instances of BRICS or other repositories or other kinds of services that might be part of that. And we're, in fact, just released a version of BRICS that supports research authentication or RAS. We're working on greater compatibility with FHIR, as well as we now support the ability. Most of our data goes into the system very highly structured using common data elements where the data gets validated, but we also have a new functionality in the meta study to actually collect and store unvalidated data. And this is one of our efforts to provide a comprehensive system, but also to support NIH's data sharing plan. And this just kind of shows you how it is kind of a data fabric or mesh. These are a number of the different instances. They share data dictionary. They have, in some cases, share a PPRL system, or they can have their own PPRL system within their system. So very flexible. In fact, we also have one of the instances deployed in the cloud, and most of these instances are deployed here locally on-prem. We support all the different kind of data types, phenotypic, imaging, and omics. These are all native to the BRICS system. So just quick summary, it's a comprehensible and flexible informatics system that can be deployed independently or in a mesh configuration. We support all the different data types, support a number of different standards and common data models, and we can deploy on the cloud or on-prem very quickly. So with that, I'm going to Dominic is going to tell us a lot more about his use of the BRICS platform and how he's enhanced the system substantially. Dr. Nathan currently serves as the Bioinformatics Director for Military Traumatic Brain Injury Initiative. He manages interagency collaborative research information systems and resources to support MTBI2 and NIH research, clinical trials, and various repositories. Dr. Nathan completed his postdoctoral training at Walter Reed, the Uniformed Services University of Health Sciences, and the NIH. He is also an adjunct professor at USIS and a research collaborator at NIH. His past non-academic experience includes developing and managing an enterprise IT system for a Fortune 500 company and managing a stroke research lab at the VA. Dr. Nathan serves on several national and international biomedical engineering advisory committees, and he is a reserve officer in the U.S. Public Health Service Commission Corps. I also want to mention that we've been working with Dominic for a lot of years, and it's been a pleasure to work closely with him and help develop the tools within the BRICS system. And he and his team bring a lot to the BRICS team, and I hope vice versa. So with that, I'm going to hand that off to you, Dominic. And do you want to go ahead and present?
Yes, thank you, Matt. I'm going to share my screen. Um, all right. Good morning, everybody. Thank you so much for the warm introduction. Is my screen visible?
Yes.
Okay. My talk today is going to be on an innovative approach to clinical trials research, specifically the NIH BRICS Unified Platform. Basically, I have no conflicts of interest, and anything I say is my opinion, doesn't reflect the opinion of the government and any government agency or department. So what is the current clinical trials landscape within the NIH? The key goal of clinical trials is really to look at applications for direct care treatment, whether it is an investigational new drug, a therapy, or a device. The objective is to find something that can be applied in clinical care. Across the NIH, there's a very diverse and rich area of clinical trials. There's everything from cancer to stroke to brain injuries. And what that has resulted in is a growing number of disparate data sources. Also, as we are moving into the era of big data and machine learning, research now is generating large amounts of data. The fact that there is a lot of data sources and large amounts of data have led to increasing burden for cost, regulatory compliances, administration, and also support. Also, there is a big appetite for machine learning and AI type tools to be used in multimodal and analytics and analysis. Um, Currently, a lot of the systems that are being used are siloed, and there are multiple systems to do different things. For example, there's a data collection system, there's the management system, there's a compliance system. And these systems also vary across institutes, across centers, and just across labs. Um, there's also a big discrepancy. Some labs, centers, and institutes have a lot of resources. They're very well equipped. And then we have some that do not have enough resources and are under-equipped. Um, a uniform solution across the NIH is currently absent. And what that means is data collection practices are often highly variable, and data curation or management is really not emphasized. And this leads to problems because it limits the return on investment for research. It also limits what discovery and validation can be performed on the data. And this is really an unspoken research problem. In 2016, there was a paper published looking at what is the effect of scientific discovery? And it's found that in a poll of approximately 1,500 researchers, 70% of researchers have tried and failed to reproduce the findings. 50% have failed to produce their own experiments. And out of the best known reproducibility analyses, psychology and cancer biology have found rates of 40 to 10% respectively. And this is a problem. And it's not an individual investigator or researcher's fault. It's just that the perspective or the infrastructure or the tools for data collection haven't really been mature or properly implemented. And we always, hear the saying by Ronald Coase out in Chicago: if you torture the data long enough, it will confess to anything. And definitely, this is not speaking on the person, but it goes back to the practices of how is the data being curated, how is it being managed, what analysis is being performed, and the appropriate documentation or the lack thereof. Also, Byron Menz actually brought up this good point. It is irresponsible to support research, but not data stewardship, because the most important commodity here is the data, and the data will survive long after we're gone. If we look at history, my favorite example is Friedrich Gauss, who came up um, with the transform to look at how to reconstruct images with a Fourier transform. But this wasn't applied until 200 years later. So if you can imagine all the work that we are doing now, there is value in the data as new methods and new applications are being developed. So what do we do about this? The NIH, being the global leader in clinical trials and research, is actually leading the way. This has been a known issue, and over the course of the last 10 years, the NIH has taken concrete steps to change and to help. So there were big discussions about repositories and collect and sharing data. And over the last year, the NIH has released a data management and sharing policy. And this has really been the guiding principle on how we should do research and manage our data. So what does this mean? If we look at the current protocol design, typically in the planning phase, we would come up with hypotheses and goals, what data is to be collected, data access, and data collection instruments. In this example, for this one study, we have two measures. One is pain, the other is anxiety. And then as we go to execution, we have the data collection, the analysis, the QA/QC of the data sets, and then the publication of the data. And at closeout, the data is often stored, but the storage is often not consistent. Sometimes it could be on a local drive, it could be on notebooks, or it could be on a network drive, and it's not oftentimes accessible. Now imagine that this is just one study, but we find a really cool finding that is groundbreaking and will change the field. And we want to validate the study. And I look around and I see my colleague has a very similar study. My colleague has also collected pain and anxiety, and I could potentially merge the data and validate my findings and make it stronger. But what is the problem? If the way I've collected my data and the way I'm storing my data isn't the same, and my QA/QC isn't the same, then merging it is going to be tedious and very time consuming, or I may not be able to even merge my data or test my hypotheses. So how do we rethink it? When we look at planning, we have to really consider a data management plan. How's the data to be collected? How is it going to be stored? Who's going to access it? And what are the regulatory compliance pieces? What are the documents that are associated with it? We also, a very important thing that we also need to have is a data dictionary. And the data dictionary really defines what we are collecting. The NIH has had a huge push for the use of common data elements. The National Library of Medicine is actually leading that, and they're curating common data elements across the NIH. And ideally, we want the data to be collected to have as much definition as possible and a reduction in free text entries, so that the data can be searchable, can be quantified, and more importantly, it can be mapped across protocols. We also want to maximize electronic data collection and reduce the amount of physical paper collections. When we look at execution, again, we want to maximize electronic data management, but also we want to come up with a standardized, as much as possible, QA/QC process that is well documented. There needs to be an audit trail as to who is accessing the data, what changes have been made, and then we have to have the ability to interrogate or query the data and provide access control to it. And lastly, the third consideration during closeout is to look at how would the data continuously be accessed, how would the data be shared, and then how do people gain access to it? So what we're trying to do is really emphasize a change in thinking during the protocol planning, execution, and closeout. And then the second part of this is we need to have infrastructure that can adequately support these research needs. So how do we come up with infrastructure? What are the options? We can look at a commercial system. There's nothing wrong with commercial systems, and there are a ton of commercial systems out there. Commercial systems offer very mature platforms, lots of capabilities, and they can be set up relatively quickly. You have some really big companies like Medidata Rave, Open Clinica, Clinical Conductor. You have a whole plethora of professional organizations that can help with the research data collection. The other option is to look at free systems such as RedCap, FileMaker, Access, any of those database systems. You can even program your own system in SQL or any of that, put it on a computer and have that in the lab. Those are relatively free and quick. But what are the downsides to these items? First of all, there is a huge security and compliance risk. If you were to go with a commercial system, nowadays the agreements are changing to a software as a service. Everything is a service. You have to pay a la carte per service, and it gets really expensive. What is also offered as a service is the data hosting, and the data is going to be on a cloud that the vendor has access to, but you may not have access. And this is a problem because it brings up concerns about data ownership, especially if funding cycles were to ebb and flow, and there's a period of funding that is not consistent, then how do you get your data back? Also, in a free system or a system that's maintained by a small lab, oftentimes we have seen this where expertise, have left, and you have a system now that's not maintained, or you have a system that's not accessible. Also, with a lot of the commercial systems, there aren't any that can really do everything from start to finish. There isn't a commercial system out there at this point in time that can do end-to-end support. What does that mean? You have to have several systems. So you have to have an electronic data collection system. You have to have a management system. You have to have a samples tracking system. And then if you're going to collate data for people across data types, a lot of these involve manual intervention, and that increases the risk or error. And that's the same thing with a free system. A lot of groups do not have the full resources to build a comprehensive system, and it's easy to oftentimes have a RedCap system for electronic data collection, and then you can have an XNet system for neuroimaging management, and you can have a different like a FileMaker or a lab matrix to manage a biosample. So now you have 3 or 4 multiple systems, and to search for one subject across these systems takes time. If you were to extract the data, you would have to merge it, and then it really creates room for error. And more importantly, knowledge retention really drops, because with the commercial systems, the moment you stop paying for them, you will lose access to the system or you lose support. And similarly with a free system, because it's so limited to a certain lab or a group, it's not easy to really share knowledge across. So what is our solution, really? We need a central NIH government solution that can provide stability and also consistency. So what exactly is a central solution, or what does this look like? These are some of the things that are needed to support clinical trials from the informatics perspective. So electronic data capture is just one small piece of it, but electronic data capture oftentimes is the most emphasized. So you can picture this as being on the tip of the iceberg, but really all the other components that you need to successfully support clinical trials. Regulatory compliance is a big item. Your system has to be 21 CFR Part 11 compliant. You need audit trails. You need to have adverse events tracked. You need a document repository, because clinical trials generates a lot of documents, from internal reviews to FDA correspondence to regulatory tracking and audits. There also has to be data management practices and also infrastructure, such as what is the data management plan? What is the data access plan? What is the security of the system? How are users vetted, and how are they controlled? How are their use being tracked? You also need study team management. Oftentimes, clinical trials involve teams that are pretty large. How do we grant people controls that are needed? There's also pharmacy to provide medications. How do you manage randomization and control? And then also we have patient management, such as scheduling, creating a master list, and then looking at appointments, reminders, and how do you manage the infrastructure that's also associated with that? So why a unified platform? In my prior slide, I talked about having a commercial system or one platform, but if you have individual systems, then you're going to end up having to get several of these to coordinate, to talk to one another. And again, the security, the compliance, and the user support is really high. Imagine I'm getting older and it's hard for me to remember my username and password. But if I had to remember my username and password to 5 different systems, that's a little bit frustrating. And then all of these systems also will reset their passwords at different times. You can imagine the administrative burden. Not to mention also that there's compliances. There are IT compliances, there are FDA compliances, there are also NIST and other federal government compliances. So these systems have to be updated and maintained, and that leads to a huge cost. In the ideal solution, we have one unified platform that can do all of these functions, and having one platform actually reduces the amount of manual intervention and the burden with user interaction, support, and also cost. So this brings us to the NIH BRICS system. So how did the system come about? Approximately 13 years ago, the NIH Center for Information Technology, NIH, NINDS, and also the DoD got together and they were discussing options because of the large amount of data and a lot of the research that was being conducted. They wanted a way to curate the data and also make it available. So collaboratively, there were recommendations on how to manage the data. That is to use the common data elements, create data management plans, and then what is the appropriate infrastructure to support these ideas. So the BRICS system was born from that collaboration. And the BRICS essentially is a secure web-based electronic system that can do data capture, storage, analytics, and sharing. It's truly an end-to-end platform. A central part of the BRICS system is having well-defined and documented data structures that conform to the NIH common data elements and the guidelines for data collection and harmonization. The system is FHIR compliant. It has that capability inbuilt. And more importantly, there are APIs that can really drive AI and ML applications. There are role-based permissions and access, and it fulfills a lot of the regulatory compliance requirements. There are also inbuilt subject management capabilities, and there's an ability to import and host any kind of data, which is nice because we can use this across the spectrum of research, from imaging to genomics to biomarker analysis. More importantly, as Dr. McAuliffe had mentioned, BRICS is unbranded and unassociated with a particular disease or organization and is highly customizable. And is also 21 CFR Part 11 compliant. And more importantly, we can pull data from CRIS via BTRIS into BRICS. And when we just a broad perspective of what BRICS is. So BRICS consists of different modules. You have a data mapping transformation tool, a data dictionary, Proforms is for electronic data collection. There's a data repository to host and share data. And then there's also the GUIDs to anonymize your data sets. You also have the query tool, which is a really powerful tool for data interrogation, merging, and searching your data sets. And then we have built the clinical trials management system to manage all the regulatory documents relating to clinical trials. We have a forum for discussions, interactions. There's inbuilt imaging tools that can manage a variety of imaging modalities such as PET, MRI, CT, microscopy. We have the training tracking module, a user management module, and then we also have a specimens tracking and management system. So this is just a high level overview of the richness of tools that are available and capabilities just within this one platform. So what did we do? We took BRICS as it was, and in a collaboration together with the National Institutes of Neurological Diseases and Stroke, we created a set of tools, resources, and enhancements in BRICS to fully support clinical trials. And we did this 5 years ago. So electronic data collection occurs via standard e-forms, but we also understand that research studies are not all the same. So there is the ability to create unique data elements or non-standard forms. And the goal is, after some time, these unique forms will become standard forms. We also have the promised library, and this is important because we have computer adaptive testing that is inbuilt. We have an inbuilt workflow for QA/QC and the ability to track what changes have occurred in the data set. We have a full audit trail, and more importantly, we have developed offline capture capabilities. So there's a mobile app that can be put on an Android, iOS, or Windows tablet. And this is particularly important, especially the NIH, because Wi-Fi sometimes can be sketchy. So we can collect data offline and upload it into the system. This is just an example of a data dictionary showing a form structure where each element in the form structure is clearly defined. So we have a description, we have the variable name, and then is it recommended, required. We also specify the ranges. So if something has a scale, it'll be, for example, 0 to 10, 1 to 10, 1 to 5. If it is a selection, we also define what the selection options are. So this helps with consistency, especially if we're doing analysis across a multi-year study. When we look at subject management, what are the capabilities we have? We can pull subject profiles. We can get all that information from BRICS. It saves us having to retype that information in. We can manage a master list. We can anonymize the subjects. We can hide the PII, PHI if needed. If a statistician or analyst is doing the research, they don't need to see the PII. If a pharmacist is looking at a medication, we have the randomization list. We can also do screening. We can control eligibility for randomization. We can blind and unblind, and then we can also do Bayesian adaptive randomization, which is a more complex type of randomization list management. We can also do electronic consenting. We can do this remotely, so we don't need the person to come to the NIH. We can consent them by sending a secure link. They can get it, it's encrypted. And if they meet the eligibility criteria, then we can consent them to the study, bring them to the NIH. It's a little less frustrating than to have someone travel all the way and say, hey, you're not eligible for the study. So we can do all the screening and consenting. We can also capture electronic signatures. So we can do this fully online. And this has certainly helped with our efficiencies, especially in the time of COVID when there was very limited entries. This is just the example interface for subject management. The minimum 3 pieces of information we need are the medical record number, the last name, and the first name. And if you could see this grayed out button here, all I have to do is click get from Beatrice, and all the other information, the birth city, demographics, home address, email, all of this would just get imported from Beatrice. So this is implemented, it's working. This is the example of the scheduler. So what we can do here is we can specify when is the scheduled visit, when is the person coming in. We have a calendar, it comes up, we have the time. We can also specify what kind of visit is it. Is it a baseline, 3 months, 6 months, final visit? We also have the location of the procedure and who's the person that they're seeing. Granted that the NIH is large, we want the ability to track visits, procedures, and at the same time, we want the ability to print this out and send it to the subject to say, hey, you're going to this location, OP5, for your headache assessment at this time, and you're seeing Dr. Hope. So we want to be able to give the person something written that's tangible, and this definitely helps with navigation. We can look up and print schedules based on a protocol, a location, or we can do it for the subject, as I mentioned prior. This is just an example of the electronic consent form. We can essentially digitize the entire form, and at the bottom we have the ability to collect wet signatures. Nowadays, a lot of people are using mobile devices, and they can easily pull this up on their cell phone or their tablet. They can sign it, and then they can return it to the study investigator. The form is often sent by a secure encrypted link. It can be sent by email or it can be sent by text message. So the subject can receive this, and we have that flexible capability to communicate with the subject by phone, to read the consent, to go through the procedures. And the IRB actually has been very supportive of this. This is the example for the samples management and tracking, where we can actually define the freezer, the storage size, the location, and it actually interfaces with barcode readers. So we have different labs that some of them have upwards of 200,000 samples, and it's easier to scan the barcode and then pull that information up. So usually the sample is associated, for example, here, this is an example of a box. So if I were to pick a sample, I can say, define what my sample types are. And then if I were to scan the sample, it pulls it up, it tells me the subject name, subject information, when the sample is frozen. And we have also added the ability to pull clinical notes from CRIS. So if the sample was procured from a surgical procedure, we can click on a button and get the surgical note for that specific procedure in here. We also have adverse event reporting, and for standardization, we have actually implemented Medra for our adverse event descriptions, and we have also included CTCAE for adverse event grading. So we get a more standardized way of quantifying our adverse events and severe adverse events. Also, with the adverse event log, there's the ability to send notifications to our medical officers if there's a severe adverse event. And these notifications are via email and also text message. Study monitoring, because we're doing clinical trials, a lot of times the sponsor would like to monitor the progress of the study. So we have our monitors come in, they can open a query, and what you see on the dashboard is if someone has a query that is open, it will say it's in progress. If it is completed, we can track the queries completed. And what this looks like is if I click on my specific question, I have an option that says, hey, there's an active query. Hence you see the red bubble. And if I click on it, I can see what is the question. So it says this question was blank. Was this intentional? And I can put a response back in. And if the auditor or the monitor is satisfied, then this can be resolved. And the beauty of this is it's all captured electronically in one location, and it's all accessible online, which is convenient for monitors who are monitoring multiple studies, sometimes across multiple sites. It could be the NIH in Baltimore, the NIH in Frederick, or the NIH across just the DMV region. It saves the person having to travel physically to look at paper copies. We have a full audit trail. Again, this is one of the critical pieces of 21 CFR Part 11 compliance. We're able to track any of the changes, who has performed the changes, the date and time. And then this is just an example of randomization. We have several different options for randomization. So typically the randomization list will be generated offline with a statistician to meet the specific parameters of the protocol. But once generated, we can import it in, and also we can control how randomization occurs. On the simplest level, it's just automated, but we have more complex controls such as eligibility list. And the eligibility list provides full flexibility to define what are the eligibility requirements for someone to be able to be randomized into the protocol. We can pull in CRIS data via BRICS for labs, clinical assessments, even consents. So this is an example of a lab that I would like to pull in. And on the top, you see there's the button Get All BRICS Data. If I were to click on that, then I get my lab results here. And sometimes there are several labs that are performed on the same day. So I can, if you see this button here, I can pull in the lab, click on it, and I can see which lab do I want in there. I can also specify a range. So there's a lot of flexibility on pulling BRICS data in. In summary, what I've talked about it, the first part of my presentation was really a change in perspective, a change in protocol development with an emphasis on data curation and data management, and how these could be built into the protocol lifecycle. Really, we want to do this to maximize our return on investment, and by looking at a government infrastructure solution that is the BRICS, BRICS platform has shown practical applications. We've implemented this across several ICs and the DoD also. And it is a mature model with a lot of good tools, such as the data dictionary, access control, inbuilt QA and QC. And then more importantly, BRICS has the ability to share data with industry harmonization practices. A lot of them have been already put in. So that isn't after the fact trying to map your data or change how it is. There's robust data management capabilities and a very intuitive user interface, and is also FHIR compliant, and there's good APIs and resources for AI and machine learning support. And this is truly a unified platform because it can support research and clinical trials from end to end. I would really like to thank my team. I would like to thank Dr. Yang Fan, who's been a great inspiration over the last 5 years with really driving this forward. Dr. McAuliffe, again, he has been a great resource. He has been with this from the start, and he's really helped this program grow. To all my colleagues that you see here, there are too many to mention, they have been a phenomenal team, a phenomenal family. We've been on this journey for several years. And I also like to thank Mrs. Sandy Martin, the data management team at NINDS, and also Dr. Avi Nath for your full support and help with this project. We also are very grateful to our users and contributors because based on the user feedback, we're now on a monthly update cycle, and we have been able to add a lot of enhancements and really change and grow the platform. And that's, again, thanks to all our users who have really helped with this. Are there any questions?
Yes, I think we have time for a couple of questions. I tried to answer a number of them online already that I thought I could handle, but we have time for 1 or 2 questions, quick questions from anybody. If you have questions, feel free to unmic and or unmute and ask your question. So I think maybe, Dominic, you could answer one question. I think that would be helpful. I think there's been a couple in the chat. This trade-off of BRICS being a centralized thing or part of an ecosystem. And I always have thought of BRICS being fitting into both of those paradigms, but maybe you could comment on that.
Yes, you have that flexibility because one of the hardest things is change. For BRICS to be a central system, it depends on where you're coming. If you're coming from not having any resources or wanting to switch resources, then that is a viable option. But if you have well-established resources and the transition is going to be too time or resource heavy, then an ecosystem with a more gentle transition would be helpful. The key reason or the key use of BRICS is, one, it's an NIH resource. It saves you a lot of cost, time, and also burden, but having a good integration plan would definitely help. So you will come out on top compared to if you were to get an external vendor or try to build a system from the ground up. Reinventing the wheel it's just resource consumption for no good reason. But if you were to take BRICS as part of an ecosystem, you will save a lot of the legwork, and then you can look at hey, what are the changes that are needed and how do you pull BRICS into a functioning ecosystem? That will be the more win-win solution because you're not starting from scratch and you get to conserve a lot of resources and also the maintenance piece of it afterwards. Over.
There's
Great.
another question from Ashley. She's asking how many extramural clinical studies use the system? What's the cost?
Extramural, within the DoD, there are approximately almost 30 studies now, and the cost is really the hosting, the maintenance, and then the data management and compliance team. So it really varies based on how many. So you, like, out of the 30 protocols, we have some protocols that are collecting 100 data elements, and then we have some that are collecting 600 to 700 data elements. So if you factor in that, it takes 2 minutes to set up one data element, that's the data manager's time. And then also depending on, the randomization list, how complex it is. So we had a Bayesian adaptive randomization list that took a lot of time with the statistician. And then you're looking at the, just the annual support for compliance and security. It is still comparatively cheaper than a commercial solution, because prior to us investing in BRICS, we did a market comparison with the critical platforms out there and vendors, and the BRICS platform became order of magnitude cheaper because whatever we have developed, we can reuse across different protocols. It's not just limited to one protocol only. And that's where the true cost savings comes in.
All right. Well, thanks, Dominic. That was an awesome talk. It's going to be a tough time competing with that talk. I really appreciate all the insights, and thanks for your time.
Thank you.
All right, so I'm going to follow up. I'm going to be a little bit more brief. We're just a little bit behind schedule, and so I'm going to go fairly quickly, but I'm going to also skip some of the slides because Dominic hit a lot of the same kinds of topics that I was going to hit, but I'll try to stop at those slides that are a little bit new, or I want to provide a little special emphasis. Okay. Oops. Okay, you've seen the team. So it's really a big team. This is Dominic's team here, and we've been working with them for a number of years. Again, this is the Sapient team down here at the bottom, and they handle all aspects of the project, the development, working as an operations team with the researchers, CDE development, and you'll hear more about that today. And then a number of my Fed team up here, and a number of the group work on multiple projects, and one of those projects include BRICS. Okay, so I think everyone knows pretty much about these kinds of things, desirable characteristics for repositories to make data fair. And I was part of this effort, and I really think about these things as I've been designing the system, and to make it a solution that really speeds speeds research and supports NIH's effort to make data fair and the new data sharing plan. And this is the details of FAIR. I think that most of you have all seen this, but we hit various aspects of this table to make data more findable. We use standards whenever possible. Our metadata standard for finding data in a study is using the DAT standard that came out of earlier. We also have make the data accessible. We use standards. We store imaging data in DICOM form, for example. We use common data elements. Whenever possible, use common data elements that we can either find at NINDS, NLM, NCI. So we try not to reinvent data elements whenever possible, but there are times, and our system supports the ability to also collect unique data. And our goal is that once we get that data, that we also make it easy for other researchers to get access to that data. It's also important for us to make our repository trustworthy. And in fact, we have a system. One of our systems is FitBur, and we're working with NEI and iGene's effort to make their repository Core Trust certified. So the FitBur system is Core Trust certified, and we're working with NEI to make their system Core Trust certified. This is how it kind of fits together. These are the community setups, the data management, how you share data, the desirable characteristics for a repository. Well, the data then has to be fair, as well as, there's a lot of effort recently about talking about how to make software fair, and to make sure that whatever software that was used to do the analysis of the data is also shared and is accessible. And then the trust principles, or the core trust principles, are all about the repository. How do you make that repository trustworthy? For example, how do you make that repository, make sure that it meets the security requirements to appropriately protect the data as needed. And then there are metrics. There are metrics to understand the usage, utility, and impact of the data and the repository. And it's important to keep track of that information so you can understand that life cycle of a repository, how long that you should keep that data, how long you continue to fund a repository or refund a repository. But we've tried to use these principles, keep these principles in mind as we've enhanced the system as we go. So lots of functionality. There has been some discussion, how much functionality do you need in the system or not? We look at it as the functionality we can provide in the system is that you don't need to connect to other resources. As a system engineer, it's incredibly difficult to take different systems and then make all those different systems work together. And it takes a long time to typically make those. But it's also important sometimes that you do need to be able to connect in those. And so we're trying to walk that line of providing a lot of functionality that's needed by the community within our system. So we can deploy a system in a month and it's going to get you, making up the number, 90% of the kind of functionality that you need, without having to try to interconnect all these different ones. But we're also working to really try to be part of the community and the ecosystem. For example, we are supporting RAS now. We just deployed our first version of RAS to NEI iGene. They were our pilot deployment. And now I think later this month we go out to all the different instances of BRICS, and they will also now be RAS compliant. But we also support kinds of different things here that we have a specimen and access tool, and we'll talk a little bit about that. We now support a lot of FHIR functionality. You could deploy a version of BRICS if you wanted that is OMOP compliant. What other? It comes bundled with a PPRL system, which is a privacy preserving record linkage system, which we call the GUID. Again, for some, it might make sense to connect to a different PPRL system, or if you don't have the funding or the time to integrate those other things, you can have it as part of this particular system. And we also have another big ability that we support all the different data types. Dominic went over much of this functionality here. One that I wanted to point out that he didn't talk about, we have this thing called a meta study. But it has two functionalities. And one of them is if you do research across the data within a repository. So I could search across the data in Fitbird to find Glasgow Coma score, total score for across 17 studies, for example. Get that data, do some analysis. I can put that data into a meta study. It gets a DOI. So we work with ODSS and generating, we went through their contract with DataCite to generate DOIs for meta studies. So once you put that data and any analysis of that data, anything else you want about that into a meta study, you publish that meta study, it gets a DOI, and then you can put that DOI back into, you can then reference that DOI in a publication. He mentioned a number of these other tools already, so I'm not going to go over those. A big one is that we really do support imaging quite well. We have a tool that helps bundle up and validates imaging data, gets submitted into the system, but then we also have a way of viewing that data. Typically, our systems have a public landing page where you can provide lots of information about what's in the repository, policies and news, etc. And then there's the access, the portal. And this is, again, controlled now through RAS authentication, and that provides you tools. But you're only given the tools, access to the tools that you've been authorized. So these systems are pretty much all FISMA moderate, and you have to go through a data access committee to get to the data. But you have different roles that you might, if you're a submitter, you have a different role and different access to different tools than if you're a data analysis user, and then you'll be given, access, for example, like the query tool. So this is, I'm an engineer, so this is my engineering drawing, and it kind of shows how everything connects. But I did want to point out again that we work with the community. If you want to use ProForms, which is 21 CFR Part 11 compliant, and we support common data elements and other kinds of standards that are important, you can collect that data and submit it into the repository. But if you're using REDCap or if you're using Open Clinica, or one of the other tools, you can actually collect data, validate that data, and have it submitted into the repository. And again, we can be deployed in the cloud, or the system can be again, we support all the different data types. Right now, none of our systems support raw genomics data or omics data. We have derived genomics imaging data as well as assessment data, but we have a workflow. That data all gets validated. It's only access to the researcher. It's private to the researcher during policy and then shared locally with other researchers as needed. But at some point, based upon policy again, the data moves to a shared state. And if you've been given access to the system through the DAC, you have access to all your private data as well as shared data. We're going to have a little bit more talk about this later about the BRICS instance on the cloud with Parkinson's disease biomarker program, or PDBP, and that's integrate or better integrate with AMP-PD data. Privacy preserving record linkage. This is something that we developed for ENDAR. It's very similar to the system used by NDA. And we're now compatible with their method of generating generating GUIDs. We have a centralized GUID system, so you can deploy BRICS locally or as a silo, and it can have its own GUID system, or it can be part of a centralized GUID system. So now we have NCATS, NEI, these other systems, as well as NINDS and aging, all connect to the centralized GUID system, so that data, regardless of which system it's in, it's all getting the same ID. Meta study, I briefly talked about. There's two methods for, I think I primarily talked about one method earlier, and that's the meta-analysis, but we also have the ability to make it pretty much act as a data store, data lake. The data you define a study within the meta study tool, and then you can upload data into that tool. It doesn't go through validation. It doesn't go through CDE checks. That data goes in, it acts as more as like a general repository. And then again, you can generate, once you publish that type of study, it gets a DOI, and then that DOI can be referenced in publications. DOIs, talked about that. This is just the workflow of our whole biosample. So you can, as a researcher, you can find out how many aliquots of a particular biosample are in or identified in our repository. Through the BRAC, you get approval and you can get access to those bio materials. We work closely with BioSend at Indiana University. So once you order that, it gets approved again, and then they ship you out the actual biosample if you've been approved. Validation and submission. So all data is validated before it goes into the system. And I'm going to talk a little bit about that. We also have a translation pilot translation tool that we're working on. It needs to be enhanced and be a little bit more robust, but that's in the progress. I'm trying to catch up, so I'm going to talk about this quickly. This is the FitBur system. 17 studies were actually submitted data about the Glasgow Coma score. So I could query across those 17 studies, aggregate that data pretty much instantly, and then do analysis. The former paradigm would be you would go to these 17 studies, they all coded it a little bit different, then you would have to harmonize all that data, and that can take a big effort depending on the kind of data that you're collecting. Where here it was pretty much instantly. It took as long as it took the query to run, which was probably 5 seconds. We also have an API to the we'll talk more about that. API access to the databases so that you can authenticate in, query the data, retrieve it, and then do some analysis. Common data elements are fundamental to our system, and it's really important, and we try to reuse and reuse data elements. This is one of the, I think, one of the more important slides that I have, where typically repositories collect unharmonized or unstructured data. Then, researchers come here, go to the database, they have to harmonize all that data on the back end. It gets reharmonized, reused, and it's quite inefficient. Where if a little bit of extra time on the front end is used, you can collect all this data consistently, validate that data. It goes into the system validated, but then you can query, as that example I showed earlier, query across these different studies and aggregate that data pretty much instantly. We use the UMLS markup language to sometimes common data elements aren't really common, but they have the same concept, and we link those together using UMLS codes. So that's helpful in that aspect, but it also makes the data more AI ready, because if you programmatically get access to the data and it has a UMLS code for those data points, you can programmatically understand that data and then reuse that data more efficiently programmatically. This is just an example of that. RAS. So we implemented the OMOP common data model. We don't have a system actually using that, but we could. We could also implement data elements for standard 5.4. I think I saw a question really quickly that we could implement those data elements, and then we would have a instance of BRICS that is OMOP version 5.4 consistent. So our system's very flexible. We don't have a standard data model underneath that we have to adhere to. Our system can be very flexible, and the underlying data model is based on the data elements, and you can define those data elements. This just is an image of the form. Here we have some imaging data just to show you how much imaging data. We have imaging data of all the different kind of modalities, over 180,000 imaging data sets. We also have bundled a tool now, VolView from Kitware, that now allows you to visualize those data elements. And we also provide metrics to better understand this. So here's a tool that we built to understand how data is being downloaded from the system. So these are data studies that have shared their data. These are data users or consumers. And then we can also see who's downloaded what data sets over time to see the value of these data sets over time. And if that data set flattens out and no one's downloading it any longer, that might be important to us to understand, well, maybe that data needs to be moved to a less expensive storage. Okay, so it's an interactive tool, very functional, and helps us, again, better understand usage, utility, and impact. And lastly, we have a comprehensible, flexible system, supports all data types, supports a number of different common data models, as well as other kinds of ways of looking at data, etc. Okay, with that, I think I'll try to answer a couple of questions. But I do want to turn it over pretty quickly. And then we might be able to also answer some of these questions online, or at the end of the talk, there'll be some more time. But, just so you know, I'm also very flexible in, reach out to me. We set up demos all the time. More than happy to talk to you about some of these. So is there any questions? I don't know if anyone's been keeping an eye. Is there any questions?
So Matt, we had a question from Anil. He's asking, does National Clinical Trials Network and NCORP, managed by NCI, NIH, utilize some of the BRICS tools and services?
No, not at this time. But again, I actually have a meeting. It's taken me a long time. It's not because I haven't tried over time. I think BRICS is starting to catch on and people understand some of the value. And I have a meeting with CBIT later this month, and my hope is some of these things will be discussed.
And Lee responded to the same, Matt, and she's saying that NCTN and NCORP, managed by NCI, haven't used BRICS. Like you mentioned, it requires over 600 clinical trial studies in the system. I wonder if BRICS can handle it.
Yeah, I can't say one would if it could. I don't I don't know that there isn't a reason why, but some of these systems, until you actually get that level, that kind of level of support, or need to be able to support that, it starts hitting the system. But we've constantly upgraded the system, improved performance, improved the design. So my feeling is we could probably meet that, but if it doesn't, we upgrade the system appropriately and to meet the particular needs of a stakeholder.
And we have a question from Toyin. Is the system intended to remain exclusive to Fed agencies long term?
I'm sorry, can you say that one again?
Toyin, he's asking, I may have missed it, but is the system intended to remain exclusive to Fed agencies long term?
What's Fed?
Fed agencies, federal agencies.
Oh, federal agencies. Okay, sorry. It's an open source. Anyone can use it. The only thing it's not open source is the GUID process, but everything else is open source. If people want to have a system, they usually contact me, and we work on setting an MOU or MOA up in place, put it in place, and then provide support for that. So last question I'm going to answer, and then I'm going to, I got to hand it off here because we're a little bit behind time. But John Prue asked, is BRICS open source? Yes. Technical documentation is also available. Okay, I'm going to hand it off to, I believe it's Olga and Henry. So it's all yours, Olga.
Yes, let me share my screen. Okay. Can everybody see my presentation?
Yes.
Thank you. So I'm going to talk a little bit how we advancing fair data principles and how we adhere to the latest NIH data sharing plan, and specifically how we do it in the BRICS data dictionary. NIH recently issued the latest data sharing plan, which came into effect in January 2023. And here I'm using a nice figure from the paper published by Gonzalez et al., which describes how one can adhere, maximize the recommendations of the NIH data management and sharing plan. And I'm talking, in my presentation, I'm going to talk about maybe first four bullets in these recommendations, which refer to how we describe the data, how we document the data, how we describe the standard identifiers, and how we use the standards and metadata, and the most recent developments we've done in the data dictionary regarding to these items. As Matt and Dominic mentioned, that BRICS is built based on the FAIR principles, and the BRICS data dictionary is built based on the FAIR principles. So we use common data elements, we incorporate standard controlled vocabularies, we provide persistent identifiers for our data elements. Our metadata is accessible and findable and visible outside of BRICS. All our data dictionaries have public-facing places. And that supports the data exchange between different systems, and that helps effectively query, find, and report the data. And as it was mentioned before, It was like 13 years of existing of BRICS, and over these years, the science made huge advances, as well as the count of the data elements also increased because it goes after the science advances, medical science advances, and the BRICS data dictionary increases the count of the CDs. So right now we have about 9,000 published common data elements in FIDBR, which means these are data elements which have data submitted against it. And we're supporting different data models and different standards. And again, the number of these models and standards growing over the years. So these all grow, which is good, but it also affects how we manage our data dictionaries, how people search our data dictionaries, how we're curating our data elements, and how we're reusing our common data elements. And that in turn affects how the data analysis and data discovery is done. And this I wouldn't call it issues. It's just the common things which all large CD repositories is facing. And technically there is nothing wrong with it, like the medical sciences doing big discoveries, they are advancing, as well as the instruments which are used to do these discoveries is also advancing. So, and we tried over time, we tried various ways how we are to organize our CDs and to help people to find these CDs and to reuse these CDs. We use keywords. We did cross mapping between different data elements from different projects. We employed referencing CDs with close semantics. We've been building ETL tool, and the most recent initiative is to annotate CDs based on their semantics and to annotate them with the unified medical language concepts and concepts codes. That's Matt already mentioned briefly, and I'm going to talk a little bit more detail about it. So when we decided why don't we annotate CD semantics, we started thinking like which terminology should we use, and we definitely didn't want to invent any additional ontology for CDs. So we started looking for terminologies, and our kind of obvious choice was to use the UMLS supported by NLM. First of all, because it's a large biomedical method thesaurus, which includes more than 200 different terminologies and vocabularies, and it links similar or synonymous concepts from different vocabularies together. It has been developed through the 30 years. And it is a tool which is used in biomedical and health informatics to facilitate interoperability across different concepts, across different projects. So it looks like the obvious choice for us. So we picked up UMLS concepts. And what we have done in BRICS, we provided the ability in the BRICS data dictionary to annotate data elements, annotate their semantics with the corresponding UMLS concepts. And we currently can do it on the data element level and on the level of permissible values. And we added the ability to search the data dictionary for the given concept. Like this screenshot shows that one can search for the concept age, and one can find the data elements which semantics has been annotated with these concepts. And this new functionality provide us with the ability to support the programmatic analysis of the BRICS data that makes our data more machine learning and AI ready, and it is greatly even more supports the data discovery. And I will talk a little bit later about it. So far, we have been mapped about 2,000 data elements in FIDBR and across different BRICS instances, but this is an ongoing effort. And among these 2,000, we mapped all core data elements for which are core for different BRICS projects. We mapped majority of the demographics and data elements, and we mapped the majority of the data elements associated with the instruments scales, which are most often used in FIDBR, in TBI, and in other research. So we did a lot of work of it. And also we, like right now, if you go to the data dictionary and click on the data element, you can see if it's been annotated with UMLS concept, you can see it on the first tab on the general detail tab. You can see this small table which shows you which UMLS concept has been assigned to this data element, and we are currently allowed up to 3 concepts, and we also allowed to create kind of compound concepts, like if the concept is which the best reflects CD semantic, if there is no such concept in UMLS, which is kind of rare, one can create the compound concept. And we also if you go to the data, to the basic attributes, and if the data element has permissible values, we provide the ability to annotate permissible values with UMLS concepts and to view these annotations through the data dictionary. You can see it through the web interface, and you can also download the data element report and see these UMLS concepts for CD and for permissible values. And we also employed use of UMLS concepts in the BRICS data discovery tool. So this tool allows to search for data across multiple BRICS instances, and one can search based on the CDE name, like you are looking for gender type across FIDBR, PDBP, CDRNS, and NEI, but one can also search by the certain UMLS concept, and also find the data elements and the data associated with these data elements which belong to this UMLS concept. So, and the last thing, here I explained what we do, and now how we did it. Obviously, we didn't want to do all this mapping manually, so we employed NLM MetaMap tool. And we first built the proof of concept as a set of Python scripts, which just digest CD semantics, hit API to a MetaMap hits API to UMLS, and then we get the spreadsheet with the UMLS codes associated for a given CD. And now we are building the BRICS module or a standalone BRICS tool, which will allow us to do it on the more user-friendly manner. And our future work regarding use of UMLS is to expand use UMLS for data discovery, is incorporate UMLS concepts in the query tool, and to enhance the BRICS UMLS to sort the data dictionary for data elements with similar semantics, which will greatly increase the searchability of the CDs. So any questions?
So I'm going to jump in here since we're a little bit behind time. But I would encourage anyone who has any questions specific for Olga to reach out offline, and she's more than happy to discuss these. I know Olga, she loves talking about CDEs and UMLS. And with this, we're going to hand it off to Henry. Dr. Henry Oji. Here we go.
Thank you, Matt. You all see my screen?
Yep, looks great.
Thank you. Yeah, so my name is Henry Ogo, and I know we're fine behind time, so I'll try to be as concise and brief as possible. Today I'm going to talk about how we have leveraged the OMOP CDM to enrich the fairness of BRICS. Previous presenters have talked about different ways that we have employed to make BRICS fair. So just for the purpose of participants who do not know what the OMOP CDM is, the OMOP CDM, I mean, basically is a community standard that was designed to structure the content of observational data to enhance efficient analysis of reliable and rich evidence in clinical data. So technically, the OMOP CDM is just a relational database schema that revolves around a person. A person here basically is a human research subject or a patient. And all the events, what we mean events here are all the data that are collected around that subject. It could be a case report form, a survey instrument, or basic clinical data that are collected on a patient in any healthcare system. Then, of course, and as you see on the screen, the OMOP CDM has a very rich vocabulary. It's one of the hallmark of the OMOP CDM standard vocabularies that are used to map all the data that are collected around the research subject. Now, if you want to look at the OMOP CDM in the context of BRICS, a lot of people ask, why would I even care about OMOP? Now, basically, the BRICS data dictionary consists of two very important object or components. One is the data element, and the other one is the form structure. With the OMOP CDM, you can basically every data set, regardless of the source, whether it's from a registry or EHR or claims data, you can map it to a maximum of 38 form structures and around 400 data elements. And very soon I'm going to explain why. So what you're seeing here is just a framework that shows the whole concept. So looking at that concept, we brought it into the BRICS platform. And how did we do that? We mapped every table within the CDM to a form structure within BRICS. So basically, we created 38 form structures, and each of them, once again, represent a table within the CDM. Then of course, we also created corresponding data element. All the tables within the CDM have their fields or columns. So each of these fields or columns were also mapped to a BRICS data element. The example we're seeing here is a form structure for the person that corresponds to the person table. Then you will see a data element in that form structure called the person ID. Then, of course, as the previous presenter talked about, every element within this instance was annotated with the Python tool that we created in-house. And we use that tool to annotate all the data elements with UMLS concept identifiers. Now, so why do we say that this enriches the fairness of BRICS? Now, first of all, the CD is structured because it's a relational database schema. It has IDs that are easily identifiable, and all the elements have also been mapped with the UMLS quiz or identifiers, so they are easily identifiable. Then, if you want to look at the accessibility part, the data, they are standardized, and if you are using the BRICS query tool, for example, as our presenters talked about, you can easily find any data elements, persons within this framework. And why is it interoperable? The CDM itself was designed to improve interoperability. The standard vocabularies have very rich biomedical vocabularies within the system. You can also trace, when you do the mapping between a source and a target, you can also use the CDM to actually track the relationship between your source and target data. Then, of course, the framework itself, the standard vocabulary, and some of the tools in it are all really usable. So we proved this concept by putting everything together. So basically, we downloaded a CMS claim data that is also publicly available. Then we developed an automated system that mapped the source data, that's the CMS claim data in this instance, to the OMOP CDM version 6. Then that was also mapped to the corresponding BRICS form structures. Then we use the BRICS validation and upload tool to load the data into the BRICS repository. Now, what you're seeing here is the general architecture that is flexible. I know that people have asked questions about can your system support OMOP CDM version 5.0? The question is yes. Majority of the elements within the CDM are reusable. And because the BRICS system is very flexible and also adaptable, we have this framework that we can easily plug in any version of the CDM, and it will not break the whole pipeline. So when you go into the BRICS system, the data that we loaded using our ETL went into a meta study, and you can use the BRICS query tool to actually see this data. You can select, you can define and select your cohort. You can merge or join tables, and you can do all kinds of downstream analytics with the BRICS query tool. Now, what is our future goal? In order to improve or enhance the reusability part of the BRICS framework, the OMOP community, they have developed very useful tools, AI machine learning analytics tools that we think we can leverage into the BRICS query tool that, apart from BRICS providing data sets that are reusable, we can also have tools, methods that can also be used or reused in addition to the data for users to do downstream analytics. So what is the take-home message here? The take-home is very simple, that now it is possible, as we have demonstrated, to stand up a BRICS instance, that is OMOP CDM enabled and compliant. With this, I would like to thank you all, the participants and the MAT and the rest of the team for this work.
That's a great talk, Henry. I think there was one question that I tried to answer online, and maybe you just can reinforce it, but and I think you talked it a little bit, but because we've implemented OMOP in 6.0, it would be some amount of work to remap it to 5.4 and deploy an instance of BRICS that is OMOP 5.4 compliant. Is that correct?
So correct.
Oh, you're on mute.
Yeah, it is. Am I on mute? Sorry.
No, not now. You're not.
Okay, sorry about it. Yes, it is correct. I don't think that it's going to be a lot of work, in the sense that the architecture is very flexible, and the good news is that, I mean, some of us are we are part of the CDM work groups that are working on the common data model and the vocabulary group. So we know that, yes, indeed, it is true that there was a pause on the 6.0, and they just want the 5.4 to get to a certain maturity level before they can do the transition. Now, majority of the data elements are reusable. On top of my head, I think if you want to look at the delta between a data element, for example, it should be less than 10%. It's time to be corrected. So, I mean, switching from a 6.0 to a 5.4 should not be a heavy lift. And once again, we already have the framework in place. It's just a plug and play.
That's great, Henry. Thanks. I really appreciate. Great talk, and I think we're going to jump into a 10-minute break. Is that right, Allison?
Yes, we are. So please come back at 10:50. We'll see you in a few.
Hi, I have a quick question. Does this recording will be shared or posted somewhere?
Yes, it will be. So after the meeting, we will send out with the email along with the slides.
Okay, thank you very much.
All right. In the hopes of trying to get us back on target, I would like to introduce our next speaker, Fatima Irfan from the Publicis Sapient team, who's going to be speaking about the work that we have been doing in support of biomedical research needs at the National Institute of Aging. Fatima, I'll turn it over to you.
Yeah, thank you, Allison. You're able to see my screen?
Yes, we are.
Great. Hi, everyone. My name is Fatima Irfan. I'm going to be talking about our work with the National Institute on Aging and our BRICS pilot instance with them. So to start off, our goal was to make it easier to find research cohorts across 4 NIA-sponsored studies. So we have HRS, the Health and Retirement Study, Health ABC, the Health, Aging, and Body Composition Study, MARS, the Minority Aging Research Study, and LOS, the Louisiana Osteoporosis Study. And the way we wanted to accomplish this goal was to undergo a data harmonization process through the BRICS NIA platform. So say that you're a researcher interested in studying the cohort of females with asthma, but you would like to look across more subjects than just your study has. With the BRICS NIA pilot project, you can look across multiple studies in a harmonized format and expand your cohort. These collaborative approaches can not only expedite epidemiological discovery, but also foster greater communication amongst investigators studying aging and promote research for topics that are not so easily addressed by just a single study. In order to start this process, first we needed to gain access to the study data, and that required some coordination with study teams and data managers. And the data was held in various formats, as you might expect. So Excel files, CSV files, .da or SAS files. But once data access was established, we had to define a common data element. And in our case, we defined a common data element to be any variable that was present in at least 2 of our 4 studies. And we looked across the study data to identify about 120 common data elements. Here you can see a snapshot of the data inclusion fact sheet, where the rows represent variable names and the columns represent studies. And a green box indicates that the study has inclusion of that variable. So this was really helpful as we went through this process. And this was an iterative process, as it required multiple phases of combing through the data documentation and the dictionaries. So the next step of this process involved grouping those 120 common data element variables into 4 form structures. So we had the NIA demographics form, the medical history form, the vital signs form, and the daily living and physical ability form. And these form structures were largely adapted from existing ones in other BRICS instances, but were further refined and made specifically for NIA. Next, we underwent a process of data transformation so that the study data could fit our common data element definitions. You can see a small example of this. As you might expect, data elements are often and sometimes collected differently across multiple studies, so we sought to harmonize them. You see an example of the same variable in 2 studies. So on the left side, you see Health ABC. The variable is do you have any difficulty pushing or pulling large objects? And in Health ABC, this variable was collected in a binary yes, no. On the right side, you see that same variable in HRS, except they collected it on a range from 1 to 6, so a scale of difficulty. And in order to harmonize these variables in conversation with the research teams from Health ABC and HRS, we decided to recode in our BRICS instance, the HRS variable into a binary as well. So there was some discussion, what level of difficulty would be yes and which would be no. So that's one example of some of the data transformations and discussions we had. And after this step, we uploaded the data and conducted validation using the BRICS validation tool, which Yep. So overall, our work can be summarized by the following ETL process. We had the data access and data definition stage. We had the create and build upon existing forms in the BRICS system stage, then data transformation, and finally data validation and upload. So currently in the BRICS NIA pilot, we have data across 4 studies and 4 forms, reflecting 1 year of study data for 16,613 participants. We can also query this data with a lot of flexibility in filtering, and in addition to that, we can download the harmonized data with a complete data dictionary, and we'll go through a demo of this using the query tool shortly. In terms of future work, we are currently working on uploading year 2 data across the 4 studies. And in addition to that, we have a separate stream where we're actually working on uploading all year 1 data across, for HRS. So that's not just the common data elements, but the unique data elements as well. This is still a pilot and all the data is remaining private, but our goal is to be able to run all the analyses for HRS year 1 in system with all the data and documentation available in a clear, accessible, and easy to use format. So I'm gonna switch over to a quick demo. This is the NIA pilot instance in BRICS, and I'm gonna go over to the query tool. So first, I'm going to query data in just one study, the Mars study. I will grab the demographics and medical history forms. I'll go to my data cart, and I'm going to join these forms. Okay. So this returned 786 rows of data. That's 1 per participant. And we can further filter this by selecting some criteria. So I'll select gender type equals female, and then ongoing medical condition types. I will select asthma and diabetes and run that query. So those 786 participants were further refined into 159. And from here, we can download the data or further manipulate how we'd like. So this is a cool example of viewing the data within a silo, but the true power of BRICS is in harmonization. So I'm going to go back and run the same query, but this time across all 4 studies. So I'll grab the demographics form across all 4 studies and the medical history form across all 4 studies. And I'll go back and I'll join them. This time we see 16,613 rows. That's across all 4 studies. And I'm going to select those same criteria from before. So in demographics, gender type, I'll select female. And then ongoing medical condition types, asthma and diabetes. And I'll run. So you see that was filtered into 1,184 rows. Overall, going from just looking at BRICS to now going across all 4 studies, we've expanded our cohort to almost 10 times the original size. And we can still trace back the data to the original source. So you see the study ID column. If I click on it, you can see that this row has come from the HRS study. But yeah, this is a really powerful tool for if you're a researcher interested in looking at a specific cohort or also just looking within your study. But yeah, thank you so much. That's all for me, but let me know if you have any questions.
Well, feel free to follow up if you have any questions in the chat, and Fatima will be sure to answer them. And with that, we're going to hand it off to Dan Gillis, and he's going to talk about the API query tool. It's all yours, Dan.
Okay. All right. I think I'm off mute now. All right. I'm Dan Gillis. I work on the FIPR imaging team, and I'll be giving a talk on the query tool API. This. Okay. So, as some background, when accessing data on BRICS, researchers want to filter data so that they can get the relevant data types that they want and filter the population group to match what they want to research. To help with this process, BRICS provides the query tool. There are currently two methods to access this query tool on a BRICS instance. One is the web-based query tool that we just saw in Fatima's presentation and earlier in this presentations. This is a GUI, and you access it through the browser. But we also have a query tool API that provides programmatic access through REST services. This presentation will be giving an overview of this API. So what is the query tool API? It's an application program interface for the query tool. In other words, it's a way for computer programs to communicate directly with the query tool. The API defines a set of services that the query tool can perform for users, and users access these services by accessing the appropriate endpoint URL. The server makes sure that users have correct permissions by using authentication tokens. These are tokens that users get after they log into a BRICS instance, and then they provide these with each request. So what can we do with the API? A lot of things. We can find all studies that use a specific form structure. We can find every form structure that was submitted for a given study. We can also query data across multiple studies and multiple form structures. We can filter these results based on data element values and perform joins across form structures. We can also download all of the records and all the files associated with the records we receive and query results. In summary, essentially, we can do everything we can do with the browser-based query tool. And I will show an example in a minute that shows some of these features. So if we can do the same things in the API that we can do in the browser, why would we want to use the API? The API provides programmatic access to the query tool. By this, I mean that data accessors can use the query tool services directly in their own programs and notebooks. Using the API in this way can extend the power of the query tool. And I've listed some examples. We can instantly create plots of the data returned. We can directly download our query results as CSVs or JSONs. We can generate filters based on the results of previous queries, and we can build data transformation pipelines and easily apply them to multiple queries, including ones that we define in the future.
Hi, Dan. Somehow you're not sharing any longer.
Really? Okay.
Yeah.
Still says I'm sharing. I'll stop and start again. Not start again, but start sharing again. Is this the last slide anyone saw?
I believe so. Yep.
Okay.
Yep.
All right. Well,
no, back one. Go back one.
Back one. Okay.
Is that right, Allison?
Yep, I think we're good to go, Dan.
Okay. So did everything with the browser-based query tool. Like I said, I went over. I'll linger a moment on the slide, but why we'd want to use it is essentially we can call query tool services directly from our own programs. And I'll show some examples of this kind of stuff in a minute during my example scenario, which is coming up. So I've provided an example scenario. So let's suppose we are traumatic brain injury researchers and we want to see how age affects outcomes from patients suffering from traumatic brain injuries. Just as an example, we use as a target metric the Gaussy total score. For those who aren't familiar, this is an integer score that indicates a patient's outcome from brain injury. And just as some preliminary research, we just want to see what kind of distribution of scores are available on FIPR. So we want to see it for all subjects on FIPR, but also all subjects between the ages of 20 and 30. So specifically, we want to do the following tasks. We want to find all the studies that have Gaussy data submitted to them. We want to find all of the Gaussy scores submitted to FIPR. We want to find all the Gaussy scores for our filtered age range, and we want to create a bar graph so we can see these distributions. We can do all of this using the API. So in the next few slides, I have some example code on there and also some output from that code, and I've outlined the output in red. As a preliminary step, we need to get the authentication token. We can do this by logging into our BRICS instance, which in this case is FIPR. We log in using RAS, and we're able to obtain the authentication token from our account page. We then pass this token into our program or notebook, and this is used to generate headers for all the API requests so that the server can validate that we have the correct permissions to access what we're requesting. The next step, we are going to find all studies with GOSI data. So there actually is a query tool endpoint that does exactly this process. You give it a form structure name, and it will give you back every study that uses that form structure. So all we need to do is send a request to the correct endpoint and give it the GOSI form structure name, and it will give us back data about those studies with GOSI data. We can see at the bottom a truncated form of what we get back. There's 17 rows. I've only showed 3 of them and only some of the columns just for space reasons, but we get back all of the data. We can see some elements we get back about them. We get the status of the study, we get the ID of the study, and the title. Next, we would like to get all of the GOSI data on FIPR. This time we use a different endpoint. This one is a general purpose querying endpoint that's a little bit complicated, but in this case, we just need to give it the form structure name for the GOSI data, and we'll give us back all of the GOSI data on FIPR that we have access to. And we can see that we get back over 12,000 rows of data. Now we want to do our age filtering on this. This is a little bit more complicated of an example. We want to join our GOSI data with the demographics form so that we can use the age recorded in the demographics form structure. We also want to pass in the IDs of the studies for the GOSI data so that we only receive results from those studies. Otherwise, the query tool will give us results from other studies with demographics data, but without GOSI data. So as input to this same endpoint from the previous slide, we'll give it the GOSI and demographics form names. We'll give it the IDs of the studies with GOSI data, and we give it an age filter that you can see sort of how it's set up on the right. We just need to give it a range start and range end so it knows what ages to filter for. And our resulting data gets us back about 2,400 rows. And finally, we will generate some bar graphs so we can see the distribution of data. On the left, we can see the overall scores, and on the right, we can see the filtered age scores. We can see that we've got significantly reduced our population, so that means our filter worked properly. We can also see that there's a different distribution of scores in the population for the filtered ages compared to the overall population of subjects with this data on FIPR. So this is just a small example, but I think it shows some of the virtues of the query tool. I've listed them out. For myself, one is that we used results from one query to generate another. We were able to make a query and then make the same query again by adding a filter very easily. And we also were able to interactively work with the data and generate plots without too much extra effort. So that concludes my presentation about this. I don't know if we have time, but if I'm open for questions and I can answer them in the chat if we need to move on to the next presentation.
I think we have time for a question or two. Okay, if not, we'll move on. But if you do think of a question, please feel free to enter something into chat. So thanks, Dan. Great talk.
Thanks.
And now we have Dr. Encinia Uma, who is going to talk about FIPR. It's all yours. But you're muted.
Can you just confirm that you see the slides?
Yep, we're all good.
All right, perfect. Okay, hi everyone. I'm Encinia Umo. I am a traumatic brain injury program director at NINDS, and I'm going to just provide you with a fairly quick overview of FIPR. So you've actually heard FIPR, mentioned a few times now, but what exactly is FIPR? So FIPR is the Federal Interagency of Traumatic Brain Injury Research Informatics System. It's a national registry for clinical TBI research data, and it integrates different data sets allowing access to high quality multi-study metadata analysis. So FIPR receives studies primarily from the Department of Defense and also from NINDS. There is a requirement from both agencies for prospective clinical TBI data to submit their data into the system. There was specific criteria in place for primarily only some of the larger TBI studies to go into FIPR, but now we've adjusted that criteria a bit in light of the NIH data sharing policy that was implemented in January. But I'll talk about that in detail in a few slides further. So FIPR also includes, again, high quality and reproducible data, such as patient characteristics and demographics, neuroimaging, clinical assessments, behavioral history, as well as varied data types. And it aggregates raw data from all investigators into common data elements, as well as we assign global unique identifiers that allow study participants to be tracked across different studies. So FIPR is really the center of the TBI research landscape. So over the years, DoD and NIH have funded quite a bit into different TBI studies, specifically into TRACK TBI, which stands for Transforming Research and Clinical Knowledge in TBI, as well as the NCAA DoD study, which looks at TBI and military cadets and also college athletes. They have over 40,000 subjects enrolled. There's also a study called ADAPT, which is a pediatric TBI trial. And there's many different, large studies, smaller studies that have that requirement to put their data into the system. And FIPR is primarily supported by DoD, NIH, of course, and we also receive support from the VA. So in terms of different data types within FIPR, I mentioned this a little bit before, but there's, of course, human subject data, including clinical, imaging, genomics. There's also neuropathology data. As of recently, about a year or so ago, there was an effort to create common data elements related to neuropathology. So those data elements are currently within the system, and there are studies that are required to submit their neuropath data as well. Several years ago, probably back in maybe 2017, 2018, there was an initiative to develop preclinical common data elements for small animal models such as rodents, as well as large animal TBI models such as swine. So those preclinical common data elements have been developed for a few years now. And although we're not accepting preclinical data within FIPR at this time, that's certainly an option for the future. So NINDS has several initiatives related to developing common data elements for different disease areas. One of those disease areas is TBI. So those common data elements go through multiple rounds of revisions, and they're also revised over the years. So we're actually going to be going through another round of revision this winter for the TBI common data elements. And FIPR actually follows suit, so that way they can stay up to date with the CDEs that's currently within the system by basically following suit for the common data elements that are developed by NINDS. So in terms of FIPR policy, I mentioned that investigators are required to use common data elements, but also in terms of data sharing, investigators have 12 months from the end of the period of performance before their data is going to be shared with the public. In terms of FIPR data access, investigators that seek data from FIPR will be expected to meet data security measures such as physical security, IT security, as well as user training, and they will be required to submit a data access request form that is signed by the investigator as well as the institutional business official. So you are certainly not expected to be able to see everything that's on the screen, but this is just a snapshot of the types of forms that's within FIPR. We have 179 TBI form structures, And we have over 8,000 data elements within FIPR. So in terms of some of the statistics, so as you can see, back in 2014, there was only 4 studies that were that were in FIPR. And now that number has grown to over 181 studies. And it's important to note that there's a difference between, studies that are publicly shared, which is which you can see is depicted in the orange line, versus studies that are within the system, but they are not publicly shared yet. And that's typically because those studies are either still ongoing or they're in their 1-year embargo period. But in both cases, you see the trend that there is a significant increase from 2014 to now, where there's, again, over 180 studies within the system, and 62 of those studies are currently within a shared state. So also in terms of the growth of FIPR over the years, there's also been a huge spike really in 2021, 2022, and so forth, where you can see that the number of records that's downloaded within FIPR has also increased substantially. And also over the years, we've really made an attempt to make the system more user-friendly. So that way, if there's anyone that's interested in accessing the data and trying to figure out, well, what type of data is actually in the system, you can easily visualize that information in several quick snapshots. So if you go to our website, you can see if you're interested in knowing, okay, well, what types of studies are actually are in the system that involved using the GOSI, for example. You can see those studies and the type of outcome measures that they collected and the similarities and comparisons between those studies. You can also see that we have over 88,000 subjects across 60 studies. Most of these subjects are males. You can see as far as our age ranges, most of the subjects within FIPR, they range from ages 10 to 19 and 20 to 29. And you can also see the different imaging modalities as well. So another component that we're tracking on a regular basis is primary and secondary publications. So oftentimes, we would receive questions that say, well, how do we know that people are actually, using the data in FIPR for any type of secondary analysis, for example, and to what extent are they using that data? So now we're actually tracking that information where we can easily tell the data that's used for primary research versus the data for secondary analysis within FIPR. So as many of you all already know of the data sharing policy that was implemented on January 25th this year, which basically said that most studies are required to find a home for their data and to share their data. So fortunately for the TBI community, we already had FIPR in place. We were actually already ahead of the curve because there were often times where I would hear investigators say in different disease areas, they would say, I really don't know where I'm going to put my data at. But fortunately for the TBI community, FIPR has been in place now for about over a decade, actually. So we already have that infrastructure in place, and it's really proven to be an excellent resource for the TBI research community. And I mentioned before that there's a policy that requires certain studies to submit their data to FIPR if they meet certain criteria, whether that's the number of subjects that's enrolled in a study or the actual direct costs associated with that study per year. But now, in light of the data sharing policy, now we've extended that to include all prospective clinical TBI studies. And we're able to manage those additional studies through the meta study module, which I believe Matt has mentioned previously. And within that module, it can contain different study types of data. For example, tabular data, software files, scripts, reference documents, and so forth. And again, the meta study is assigned to a DOI that can be cited for journal publications as well. And lastly, I want to mention that NINDS is funding a biofluid repository, and it's receiving samples from multiple TBI studies. And the great thing about it is that we are coordinating the catalog with FIPR to allow investigators to search for the different samples that's within BioSend, and also to take it a step further and to order those specific samples that's within BioSend. So it's great to see that, you know, the collaboration really between FIPR and BioSend, and really a benefit for the research community, because not only can investigators, you know, use or access the data within FIPR, but they can also have an opportunity to have access to the actual samples within BioSend as well. And then the last thing that I want to mention is that there's also an effort to expand FIPR even more by including polytrauma data. So including trauma data in addition to traumatic brain injury. And that is primarily being supported by our DoD colleagues. So with that, I will end there.
That was a great summary, Ancini. I really appreciate that. I think we have time for a couple questions if somebody has any questions. And as always, feel free to reach out to us either through chat or offline to Ancini or any of us, and we'd be happy to answer your questions. But with that, we're almost back on time, Ancini. Thank you.
No problem.
And next up on the list is Andrea Lutz, who is going to talk about the PDBP use case.
Awesome. Thank you, Matt. As Matt said, I'm Andrea Lutz. I'm one of the PDBP project managers for our Parkinson's disease data management resource. And I'm going to be presenting a PDBP use case today. So, all right.
And we do not see your screen.
Let me try again. I think Dan had that problem too. How about now?
Yep, looks great.
Okay, great. Well, here was the intro slide. But yeah, I'm going to be going over who's involved in our program, our PDBP DMR stats, our data collection process, and our biosample request and order process. You might hear me throughout the presentation refer to PDBP as PD, a little shorter, and then biosamples also referred to as biospecimens. So just to note. Okay. Our program consists of PDBP investigators and their clinical coordinators and research nurses. It also includes our PDBP program and review staff, our operations team, and our biorepositories. We have 342 active users from 26 different countries. We have over 56,000 records submitted this year, and we have over 62 million records downloaded this year. Our study population includes Parkinson's disease. It also includes dementia with Lewy bodies and some others you can see listed, as well as healthy controls. Not only do we collect clinical assessments in our database, but we also collect corresponding biospecimen or biosample data as seen here. Though our users utilize all modules in the PDBP DMR, our clinical coordinators mainly collect data through our Proforms module. In this diagram, you'll see here at the top, we have our clinical coordinator. They'll collect those clinical assessments and enter that into their study protocol in the Proforms module. And then from there, that clinical assessment data gets submitted to their study in our data repository module. Now let's take it back up to the top, because while clinical coordinators also collect those clinical assessments, they're also collecting corresponding biospecimen or biosample data. From there, they'll ship those biospecimens to the BioSend biorepository at Indiana University. Now, the data repository module in our system and biorepository system connects through API to populate our query tool, and it populates it with the biosample catalog, which brings me to our next slide. And so this is what part of the catalog looks like here. So if an investigator is interested in ordering any of these samples, they'll submit an online application to our Parkinson's Disease Biospecimen Review Access Committee, or PD-BRAC committee. So how is that done? What's kind of the process through that? First, they'll of course need to make sure they have a PDBP DMR account. They'll then use the PDBP DMR to search for that sample availability in our query tool. Next, they'll submit an online application through our PDBP public site. The applications are submitted through an online web form. You can see here on the slide, these items are required for the application. And then third, the PD-BRAC will review that application. Applications for biospecimens are received on a rolling submission basis, and they're reviewed every other month. So an investigator will either be notified by email of whether or not they're approved upon revisions, or denied. In this case, they were approved. So next steps. Once approved, we'll hold an onboarding sample distribution meeting. This meeting will go over things such as how they want their box designed, when they want shipments done, or how they want shipments, where they want that sent, MTA. Then we'll hold a PDBP biosample order demo call. So this call will show them or answer any questions or additional questions they might have about the query tool and sample selection. And then it'll also demo how they would go about ordering the samples officially. Okay. And this is how the investigator would go about selecting and ordering samples. So first, they'll navigate to the query tool module. They'll then select the Forms tab here. In the search field, they'll type in catalog and select search. That pulls up our catalog here. To view the data in the catalog, or the samples that are in the catalog, they'll want to select this larger data cart, and that adds it to our Step 2 Refine tab, or Data Carts to View. Down here, I have some other forms. If investigators are interested in any other clinical data, they will add those to the cart as well. They'll add it and search for it in the Forms section and add. You can see here in this example, I've added a demographics and an MDS UPDRS form, and then I've done a form join here to combine the data. Then they'll want to run the query. I want to point out an enhancement with the form join process that we implemented in the PDBP query tool that I feel has greatly improved the biosample filtering process and selection process, not only for our users, but myself as well, helping those users. So you'll see here, previously, when joining on Forms in Query Tool, the Query Tool would join on this subject GUID only. Because of this, users would have to manually make sure they were associating the correct clinical type with the correct clinical assessment and sample time points, excuse me. So you can see here, it would match the 12-month with 24, and then they would have to manually manipulate on their end. Now the system joins on GUID and our standard PDBP visit type, removing that issue of unassociated clinical and sample data and that need for that manual data manipulation. So it's greatly improved the process and sped that up as well. Now, let's get back to applying filters. We kind of saw this with the other presentations. Typically, when you're joining a lot of forms together, you're going to get a lot of rows of data and a lot of samples that you're most likely not approved for. So we need to narrow this down. And to do so, you'll just apply filters to the columns of interest. It could be gender, it could be just different cognitive scales, etc. And so I've done that here and narrowed down my selection to 247 rows of data. So from there, you can either go ahead and order those 247 rows of data, or you'll download that amount to a CSV and continue your additional analysis offline and filtering to get it down to your approved amount. So let's say I wasn't approved for 247, I was approved for 100. I would most likely do this option here. And that's typically what does happen with our users. But Query Tool filters down majority of that and does most of that work. And then you do the rest offline if you want to kind of refine further and do things like that. Okay, so, right. Once you've determined your final samples to order, the investigator simply goes back into that Query Tool module. They only need to add the biosample catalog to their data cart to view. They don't have to add the other forms. And they can navigate to this orderable biosample ID column, apply a filter, and then the query logic box appears, and they can simply copy and paste all these unique orderable biosample IDs from their CSV into this field here and run the query. This narrowed down, in my example, the sample selection to 100 rows of data or 100 samples that I want to order. From here, I would add those selected samples to my cart. You have this notification that appears that says visit your queue. You'll click on that blue link there. And then this takes us to our biosample order queue. The order queue is where you can update the number of aliquots you want. Sometimes investigators request 2 or 3 aliquots, or they're approved for that. And so they would update that here in mass. And then they would go ahead and select all their samples to add to their order. Adding to the order will take them to the order form, which looks like this. They'll need to add their order title. They'll need to upload their supporting documentation. For us, it's typically their approval letter, nothing more. And then they'll need to enter a shipping address and any special instructions for BioSend, who will be shipping these samples to them. Once they're finished, they'll select this Save and Submit button. Say they're not finished and they need to go back and check something, they can just select Save and Exit and come back to this form and review and then save and submit their order. All right, from here, a DMR admin, such as myself, will get an email saying that an order was submitted. We'll go into the system, we'll review, approve, and submit that order through the DMR. From there, BioSend team will receive and prepare that order on their end. They'll then ship those blinded samples to the researcher. The researcher will receive those samples, they'll perform their analysis, and they'll eventually submit their results back to the PDBP DMR, which is another process for another time. And to date, we've had 108 distributions to 52 separate investigators, totaling over 34,000 sample aliquots distributed and counting. We have a lot currently in the works now, and it's always ongoing. And then here, this is just a graph that's available on our PDBP public site, along with some other data graphs. So I recommend taking a look there and checking out what sample availability we have. And if interested, definitely apply for a PDBP DMR account and take a look. And then these are just some of the publications from our contributing investigators. And with that, I will take questions.
Yep, I think there are a couple minutes or time to take a couple questions if anyone has one. Great talk, Andrea. Thank you.
Yeah.
Well, not seeing that there are any questions at the moment, and we're a little bit behind time. I'm gonna go ahead and hand it off to Christine Treese, who is going to talk about the PDBP Google Cloud migration. Thanks again, Andrea.
Of course.
Thanks, Matt. Thanks, Andrea. Okay, let me share my screen.
We do not see it yet.
Yeah, sorry, it's still loading. Give it a second.
Okay.
How about now?
We're all good.
Okay. Just put that in presenter mode. Okay, everyone can still see it?
Yep.
Great. Okay, thanks, Andrea, for kind of kicking us off there with PDBP. I'm going to go into a little detail about our PDBP Cloud Initiative. My name is Chrissy Treese. I'm also a project manager on the PDBP project with Andrea. And we moved PDBP to the cloud almost a year ago, December of 2022. And this was the first BRICS instance to make this jump. So I'm going to just talk a little bit about that initiative. So to start off, a few motives for moving to the cloud. A big one was, NIH encouragement to start moving all projects to the cloud. And I think this really has to do with that for, allowing for future enhancements to PDBP's scalability and performance. Another larger motive, and probably the reason that this was the first instance to move to the cloud, is that we're preparing for a rather large AMP-PD data merge. And I believe the next slide, I'm going to talk a little bit about AMP-PD. So AMP-PD is the Accelerating Medicines Partnership for Parkinson's Disease. This is a program that is a public-private partnership between NIH, biopharmaceutical companies, life science companies, and nonprofit organizations. It's managed through FNIH, and it currently holds about 2 terabytes of data that are already housed in the Google Cloud of Parkinson's disease data. So the future plans is really to merge all of this data into PDBP. So before we even started development, obviously for moving to the cloud, we did kind of an extensive cloud services analysis. Our BRICS team, we interviewed Google Cloud and Amazon Cloud support teams. We had multiple meetings and discussions with both teams. We prepared a full comparison of the 2 platforms. And really tried to weigh both options based on PDBP's needs, their specific needs. And after completing our analysis, the NIH PDBP team was able to select Google Cloud as the best option. This was mostly due to cost support, available support that the 2 platforms offered, the enhancement capabilities for the future, and the ease and cost of data transfer. So again, like I said, mentioned before, that AMP-PD data set is already housed in Google Cloud, so that played a big role too in our selection. And then we also noted here that this was deployed under the STRIDES model, and the STRIDES team played a large role in supporting this initiative. So here's a little bit on our cloud development. So, I won't go into detail here, but this kind of lists off all the real basic steps that went into place for all development work, infrastructure work, and configurations. We noted all the different stories here that are listed that were STRIDES supported, and that STRIDES help support each of these pieces. Scott's also on this call too, who's our infrastructure expert, who could elaborate or answer any questions that are specific around the development work that went into moving PDBP to the cloud. And then last year, December 2022, we successfully deployed PDBP to the Google Cloud. This was a lift and shift operation, so enhancements are planning to come later. It included 3 different environments: stage, UAT, and production. And this was roughly about 6 to 7 terabytes of data. This transition went really smoothly, and we had strong support from the STRIDES team. Now this slide, so I did want to take a little bit of time to talk about ANA. So ANA during the when we did move to the cloud and we were having those when we had those initial discussions about moving to the cloud, we started to investigate how this might change our ATO, our authority to operate. And during those conversations, we were made aware that we need to do a full review, a full ANA review, which is assessment and authorization, before we can, well, not before, but during this whole process in order to maintain our authority to operate. So ANA, this is a big comprehensive assessment. It evaluates system policies, security components, documentation, vulnerabilities. It's based on the NIST Rev 5 framework, which I have a link here too, if anyone is interested in seeing that full list of control families. There's about 20 different control families with multiple controls under each control family, and each one needs to be addressed during a full review. The BRICS team, this full review is for the full BRICS platform. It's not specific to PDBP. And we are in the process of a full review. The full review happens every 5 years, and then there's smaller reviews yearly in between there. And again, this is something that is required in order for us to maintain our ATO, our authority to operate. I will point out that we are still in this full review, and even though we went to the cloud last year, we were doing the full review last year using the Rev 4 framework, and then they updated early this year to Rev 5, which required everybody who was undergoing a full review to basically start over using that Rev 5 framework, which is where we are now, and hopefully close to completion. So I put, there's 2 basic stages to ANA. There's the assessment stage, and then there's that authorization stage, which is that final authorization decision, and that final ATO is received. And then I just really wanted to end on the note that this PDBP Cloud initiative has really laid the groundwork for the accelerated deployment of future instances to the cloud. So we were the first. It went rather smoothly. And a lot of these pieces that we use to move PDBP we'll be able to piggyback off of for any future instances looking to move to the cloud. And that's it from me. Any questions?
You might be on mute, Allison. We still can't hear you.
Yeah.
There you go.
Chrissy, thank you again so much for your presentation. Definitely appreciate hearing about the PDBP use case. So thanks, Andrea, and thanks, Chrissy. Looking at time, I want to first and foremost thank all of the presenters today. We had some really exciting content to share with all of you. So thank you to the presenters for the time, for creating the slides and sharing your stories today with the audience. And I also want to thank the audience. Thank you so much for making time today to attend the BRICS Working Group seminar. I would like to just kind of close by kicking off a Menti poll. So 2 ways to access it. You can use your phones and access through the QR code. Also, the URL is being presented as well as the access code so that you can join the Menti. So I'm going to give everyone a minute just to figure out how you'd like to join, and then we'll kick off the Menti. Just maybe a quick show of hands, thumbs up. Are people able to access the Menti? Okay, are we ready to kick over to the Menti? Or are folks having issues? It looks like some hands raised. If you want to come off mute.
Yeah, I use a QR code. It worked fine.
Awesome. Thank you for the confirmation. I'm also just going to drop the URL in the chat too for folks. That would be helpful, I think, as well. So just give me 1 second there. All right, well, hopefully we can kick it off. I will share my screen. We have a series of questions. Definitely love to hear your feedback. So 1 second while I share. Can everyone see the Menti?
Yes, Allison.
Perfect. All right. So first question, which BRICS module would you be interested in learning more about in a future meeting? Please feel free to select all that you have interest in. And if anyone's having any issues, feel free to drop them in the chat and hopefully someone from the team can help you with troubleshooting. Okay, about 25 seconds left. I don't know, Matt, if you want to just jump in and start to provide any feedback on the results that are being displayed.
No, that's just great to see that just kind of a broad interest in a lot of tools, but it's neat to see that there's a strong interest in the data repository and the query tool. So yeah, great.
Great, perfect. Jump to the next question. Which additional features or capabilities would you be interested in learning about? So the last one was really specifically about the modules that are built into the system, and this is more about the features. Again, looks like we're getting an interesting distribution across different features and capabilities. Okay, does anyone have any questions specifically about any of these features? If you do, feel free to drop them in the chat. We'll try to address them. I'm going to close voting. So if you have not voted, please go ahead and vote or share. Okay, we're going to jump to the next question. Next question is, do you use CDEs in your research?
Allison, your question, yeah, it's back.
Okay, yeah, sorry. 2 screens. And if you're not aware, CDE, we've heard it many times, common data element. So And if anyone's willing to share, if you are using CDs and it's been beneficial, if you want to drop that in the chat, that'd be awesome. If you are not using CDs, is there interest in learning more about CDs? Obviously, we are happy after today's seminar to take any of the presenters are happy to feel free to reach out to them offline and ask any questions you may have, as well as to Dr. McAuliffe. Right, so we're going to close voting for this question and move on to the next. What is the greatest roadblock that you feel is impacting your ability for data sharing and reuse?
Oh, what is the
Give people a couple more seconds to add in your responses. I know it was a long list, so definitely read through and please select your greatest roadblock. Okay, I'm going to close voting for this question so we can move on to the next, which is, do you use any of the following in your research program currently today? Thanks everyone for replying. I'll give everyone a few more seconds and then we'll move on to the next question. Okay. Now this is a free-form response. So we're asking, what other features or data types would you like to see included in BRICS in the future? So please, if you could just reply with a single word, that would be most helpful. Again, what other features or data types would you like to see included in the future? Give everyone another 10 seconds, 15 seconds. Thanks for sharing. Please keep dropping them into the Menti. All right, awesome. Thank you. We're going to close and head to the next question. Please share specific examples or case studies presented during today's seminar that resonated with your research goals or projects. Awesome. Thanks for the responses. Please keep them coming. I know we're getting close to lunch hour, and I appreciate everyone who's been here with us for the past few hours. So definitely final stretch here. Thank you so much. I'll close the question in 15 seconds. Right. And the next question is, what emerging trends or technologies do you think will shape the future of the bioinformatics field? Please feel free to select all that apply. Awesome. Thank you, everyone, for the responses. I'm going to close out this question, and I don't know, Matt, if you want to jump in and close this out since it's 11:57.
Yeah, I want to thank everybody, all the presenters, as well as everyone who was able to attend. I think really, really good feedback that's provided. We loved the questions. And with that, if you have additional questions, please reach out to me, and we'd be more than happy to answer those and or provide some demos. So thanks everybody, and have a great rest of your day.
Thanks, everybody. This is really helpful.
Yeah, I agree. Thanks, Matt. Thanks, Allison. This has been fantastic.